very clear that the ethyl acetate was always going to pass, and the pores were just too small to squeeze water through. So we really needed very high pressures, very, very high flow rates to even get a chance of getting any water through. So our flow-based properties of our membranes are related to the pore size. We can control that using the porogen content in the 3D printing process. So that's the first part of work where Harry has been working on hydrophobic membranes for phase separation, where we combined a fluorinated monomer to get a hydrophobic membrane with a porogen to get a porous membrane being formed, and we used it to phase separation. The next one we'll be looking at is using the same print pulse print method, but then for the electro extraction of DNA. This is the setup that we had in mind. So what we really wanted to do is have a lysate of E. coli. So I hope I'm now going to make my biotechnology friends here a little bit happier. We had E. coli in there, a lysate of E. coli with soup, because we wanted to extract the DNA from an E. coli lysate for amplification by polymerase chain reaction. We'll be talking a little bit more about DNA amplification tomorrow. Polymerase chain reaction, or PCR, is an amplification technique where you can multiply your target DNA to a level that you can more easily detect it. So this is the device that we really wanted to have. If we look at the cross-section here, our aim was to have an E. coli lysate where we've got popped E. coli cells that spat out their contents, their proteins, and their DNA. We put some buffer in a receiving chamber, as well as in a chamber where we put the electrode. We apply a potential difference. We use this as a size mobility trap. We've got large pores here that allow for the passing of the DNA, but not of the proteins and the cell debris. We've got very tight pores here. So that this reservoir in here, DNA doesn't go in there, but we still have got large buffer capacity in order to deal with the electrolysis products. Remember what we saw in the free flow electrophoresis devices, that we need to have, if we want to do electrophoresis for an extended period of time, we need to have a large buffer capacity. But we didn't want to have a large receiving chamber, because that would be diluting our DNA and increasing our detection limit. So here we've got the best of both worlds. We've got a huge reservoir here with our buffer, so good buffer capacity, no pH changes. And we've got a very small reservoir because we also took the floor up a little bit. So we've got a very small reservoir here, so we can concentrate our DNA in our size mobility trap here created in our 3D printed device. If we think as an engineer on the numbers, what are the materials that we need to have in? To create our size mobility trap, we would like to have a microporous membrane with a pore size around 300 nanometers to allow our DNA to get through, but not our proteins. The other thing we would like to have is a mesoporous membrane that's got a pore size of 20 nanometers, or even smaller, to allow small molecules, small inorganic ions to be moving through so we can desalt. They need to carry the current, so we want to have a little bit of ion transport, but we don't want our DNA to be ending up in our electrode reservoir. So a chamber to pipette our E. coli lysate into, collection chamber to have the DNA going through, and our large buffer waste chamber where our ions, small inorganics that are being transported by the field end up, and that has the big buffer capacity. To get that, we need to optimize the resin formulation. To print that in one print process, we have another problem that we need to address, or another restriction. And that is when you print by a print pulse print approach, every layer that you print needs to have the same exposure time, independent of what material you're printing in. So one print run has got one exposure time. If you do a resin exchange to do print pulse print, you cannot change your layer exposure time mid-run. So all your layers, your different materials that you're combining together, need to have the same exposure time. So that's one criterion in optimizing our resins we needed to keep in mind. If we overexpose one of the layers, even if we would be able to change that exposure time, we could probably fill up voids that we'd made previously. So there is no need to start hacking the printer and get to a longer exposure time, because it will destroy the rest of our prints. Something else that we found out is that porogen-containing resins require about 4 times the exposure time in comparison with normal resins. So we'll need to do some playing around and fiddling around to get a normal resin combined with a porogen-containing resin to integrate those two materials. So porous properties, exposure time, were all optimized for this work. So first, we started looking at what is actually the effect of the exposure time. Even if we may not be able to use it, what happens if we change the exposure time? So here we've got different layer exposure times, 30, 60, 90, and 120 seconds. Can see when we look at the material, so here we've got the pore diameter, and here we've got the amount of flow we get through in our perimeter. The more flow we've got, the more pores of that size we have. So we can see that with a long exposure time of 120 seconds, we got small pores. The shorter the exposure time, so here we've got 30 seconds, we've got larger pores. So we can, in principle, use the exposure time to change the pore size. And as we saw on the other side, the density increases when we have got a longer exposure time because we get more monomer being incorporated. Our porosity decreases. Again, we get more extensive polymerization, more branches being formed. We get a more dense material. That is also confirmed when we look with the electron microscope, when we can see here we've got a 30-second exposure time, 60, 90, and 120 seconds. We can see with a low exposure time, we've got at a 1 micron scale bar and a 200 nanometer scale bar, we've got wide open porous structure, typical to a polymer monolith. When we've got short exposures time, the longer we expose for, the more dense the material that we form becomes. So we can play around creating different porosities using the exposure time, even though in our printer that wasn't going to be the best thing to do. And we really liked the nice pore size distribution for the short 30-second exposure time. Next thing, we actually wanted a little bit smaller pore size. So the next thing we could do to get more porous structures being formed in that short exposure time is to play around with the photoinitiator concentration. The target that we had was about 300 nanometers. We looked at 1%, 2%, 3%, and 4% photoinitiator. And we can indeed see the more photoinitiator, the more radicals we get formed, the narrower the pore size that stays out there. Again, we can confirm that with SEM, where we can see that with 1% photoinitiator, we get a relatively open mass of cauliflowers being formed. The more photoinitiator, the more dense our cauliflower becomes. So we can play around with the photoinitiator. Once we've got our photopolymerization time set, so we've got our layer exposure time set, we can change our porous properties of our resin, adding more or less photoinitiator. So we were very pleased about that. And again, more photoinitiator, we get a more dense material and a less porous material. And we liked the one for our first membrane. We wanted to have a membrane with a pore size of around 300 nanometers. We really like the ones here with the 3% photoinitiator concentration, because the other ones, if we went, even though we got a smaller average pore size, it's a really broad pore size distribution, which means that a lot of our macromolecules would be still sneaking through. So we selected here the 3% photoinitiator. So this was the polymer monolith that we decided would be really good for our membrane, separating the E. coli lysate from our DNA collection chamber. But we now played around with a chemical toolbox, we thought reasonably well. Our pores were nowhere near 30 nanometers yet, so we needed to get a new trick off the shelf. And that trick was to change the amount of porogen we had present. The porogen is the thing that holds that creates the voids. So the next thing that we did was decrease the amount of porogen. In our standard resin, we had 70% of 70 volumetric percent of porogen being present. We dropped that to 50%, and we ended up with an average pore size of about 20 nanometers, still with a very nice distribution. We dropped it even further to 45% volume percent. At that point, we still got a material being formed, being printed, but when we filled both sides with buffer and we applied a potential difference, we did get a current. So the pores of the material printed at 45% porogen were too small to allow even for the transport of small ions, which means that it was not going to be a material suited for practice. Materials all optimized, Harry's part of the work had been done, and this is where the project got transferred to another student, who's Sumin. So Sumin is Korean and is doing her PhD at the moment. And she's doing her PhD on sample preparation approaches for DNA lysis and extraction, and she's the master of PCR and other amplification reactions. So Sumin jumped in here to help Harry characterize and optimize the conditions, because all the samples that we extracted with the DNA here in the reservoir, we needed to amplify them. And we needed to get the lysate as well, and Sumin also does all the culturing of E. coli. So we got Sumin involved, she got E. coli samples, she got little bits of our extract chamber back, she took it to her PCR machine, and she did all the number crunching of the slides that you'll see later on. So we've got the mesoporous membrane. So the first thing we did was the optimizing of the extraction conditions. The parameters that could influence that we could think of were the time that we used to do the electroextraction and the potential that we applied. So we applied potentials between 2.5 and 15 volts, and we did that over times ranging from 5 to 20 minutes. By doing those experiments, we found out a couple of limitations. When we applied our potential at voltages higher than 12.5 volts, so 15 volts or so, we got a significant EOF being formed, supported by our membranes, which led to the pumping of liquid from one reservoir to another, which led to significant differences in height. In addition, the membrane that was pumped empty started evaporating and drying out, which is not great for electrophoresis either. So we decided to keep 12.5 volts based on those observations at our maximum voltage. We also found that if we did the extraction for longer than 20 minutes, our small chamber where our extract was collecting actually started drying out because of evaporation as well. The total volume was probably 20 microliters in an open chamber, getting warm because of the electrophoresis taking place, a little bit of Joule heating, and the volumes became smaller, irrepeatable, and our error bars went on. So we defined our optimum conditions, getting a good extraction yield at 20 minutes and 12.5 volts. Here we've got that plotted as the CQ value. This is where my biotechnology class says, yes, of course, that's great. And all the chemist says, but it's the lowest bar. CQ value is the number of cycles it takes in PCR to get above a threshold and you can detect your amplification products. If your CQ value is low, it means that you detect your amplification project after very few cycles, which means that you've got good sensitivity or a large amount of DNA present in your sample. If you've got a very low amount of DNA present in your sample, it takes more cycles, so your CQ value goes up. So unlike in analytical chemistry, where a small peak means low sensitivity, and the higher it gets, the better it gets, in PCR, we need to think the other way around. The lower the CQ value, the better. Takes a little bit of rewiring around, figuring it all out. But here, our optimum is actually the lowest point, not the highest point. So if we look at the recovery rate here, we can see that under the optimum conditions, 12.5 volts, 20 minutes, we got actually very close to quantitative recovery of our DNA. So we could extract almost all the DNA present in our sample chamber into our acceptor chamber and go to PCR. At the same time, we looked at the extraction efficiency relative to a kit. And we can also see that we were at 94% of the efficiency of a kit. We did statistical analysis on this with the arrow bars, and this was not statistically significant. So the efficiency of DNA extraction of our little 3D printed device was equivalent to the spin columns that you buy to do DNA extraction with, using traditional laboratory techniques. The other thing that we quantified that is not on the slides is that we quantified the amount of proteins present in each of the reservoirs using the nanodrop. And we had a large amount of protein present in the sample, which you expect in an E. coli lysate. We could find less than 10% of protein in our sample that went to the DNA amplification. No protein in the buffer reservoir at the other end in the acceptor chamber. We also could not find any DNA present in the acceptor chamber, meaning that our membranes were effective. We could get DNA, but no protein through our first membrane. We did get small inorganic ions, but no DNA through our second membrane. So proof to the pudding, can we actually detect cells, E. coli cells? So we made up different cell concentrations, added the lysing agent, and here you can see if we get more cells in our solution, our CQ value drops. But when we only have got 1,000 E. coli cells per microliters present, we still get a respectable CQ value of 28. People get only concerned about sensitivity if your CQ gets over 35. So we can use our device to quite safely detect reasonably low levels of E. coli. So that's our application on DNA. And we've looked at changing a little bit of our chemistry and a little bit of mainly of our chemistry for optimizing the porous structure. Next bit of work, we focused on looking at the layer number and the layer height in order to make porous materials. And we applied this to chemotexes. So biotechnology people, now we get really excited. We're moving away from E. coli and we start looking at, who are the guys in the middle? Zebrafish. Who is our little critter sitting in the corner? Planaria. Never heard of them before. They're little critters that swim around in the mud on the bottom of rivers, and they're a very good indicator of biological health or ecological health of the systems. So here, this little cheeky guy was going to be introduced in a microfluidic device, and a couple of these zebrafish as well. The experiments we wanted to do were chemotexis assays. Chemotexis is if I like some food, I get close. If I don't care, I kind of randomly walk around. If I hate some food, I move away. Very simple phenomena. So what we wanted to know is if we could use some microfluidics for chemotexis, can we get our little critters, our zebrafish and our planaria, can we use a microfluidic device to do some chemotexis testing to see if they go towards a food source? So this is the device that we wanted to use. Having a little chamber for them to be swimming around in, with having two different membranes, one side where we could have a blank solution, the same of the media that they were going to be swimming in, and one where we could inject a chemotractant to see if they would be moving towards the chemical that we added. Again, we used the print pulse print system, where we first print part of the device, exchange the membrane to add the porogen containing resin, print the membrane, go back to print the next part of the device, printed the second membrane, printed the final part of the device. As you can see here, the membrane, similar material, and if we look at the 5 micron SEM, we can see that we've got our familiar cauliflower-like structure, typical for porous polymer monoliths in there again. So again, we're printing a material. So the resin exchange methods was used, and we looked at the global structure and did the characterization. This time with the membrane features, we actually wanted rather large pores because we wanted free diffusion of our fluorescein through our membrane to enter the cell as a chemoattractant, we actually only want to use the membrane to be a barrier so that we could do our fluid manipulation and exchange different solutions and have a container. So around 400 nanometer pores would be good enough. But it was important that our membranes were going to be robust because they were going to be used by people not used to handling microfluidic devices. This is the point where we looked at the printer settings, how we could change the porous properties. So we looked at different layer heights. So we've got a layer height of 200 micron, which ends up with relatively fine pores, around 200 microns. If our layer height is 150 micron, our pore size gets a little bit bigger to 300 nanometers. If we have 100 nanometer, we get our 400 micron plus pore size. One of the consequences also of reducing the layer height is that we lose some of our structural rigidity. So we get pore size distribution and the average pore size increase with decreasing layer thickness. The porosity of the membrane increased and the density and strength decreased, which led to cracking, incomplete formation, and shrinking. No, that's the number of layers. So we've got the layer thickness here, and then we've got the layers. These membranes were rather fragile with a really big pores, so we did decide to use 3 layers of 100 micron thickness in order to get the right porosity as well as strength of these membranes. So the experimental setup, and this is for me as a chemist where things get excited. We get living things into our chip. So Aaron, a colleague of mine, helped us with the husbandry of the zebrafish. So they made little babies and we could introduce them into the chip. First, we looked at fluorescein as a dye, and here you can see when we started, no fluorescein was present in the chamber. We get diffusion taking place, a little bit of convection. You can see diffusion is not perfect. And here, after about an hour, we can see the whole chamber being filled with fluorescein. That's not a situation where we want to be in to look at our chemotextus experiments. So this says that about 30 to 40 minutes, we can do our chemotextus experiment, and after that, our operational window is gone because the whole cell will be filled with our small molecule chemoattractants. So here we've got our little beast. So the little dots that you can see here, they are our zebrafish. Here we've got a little glass tube that we can allow us to pipette the zebrafish embryos in, and then carefully take it out to get them to swim wherever we want when we are ready to start the experiment and we've got the camera running. Here you can see the zebrafish just after the vial here has been taken out, and they're reasonably evenly distributed. This is where they start thinking, ha, I can see food. I get closer. So you can see that they're all moving to the top of the line. And here they are still kind of at the top towards the end of the experiment because that's close to the food source. So we can plot that here. This is where we see more of our zebrafish over time. The blue is where we see fewer of our zebrafish. And here on our heat map on where are our zebrafish embryos, or now our newborn zebrafish. So they're not the embryos, they're the little ones, the little critters that can already do some swimming. So we can see here that the zebrafish spend more time close to the chemoattractant than they spend away from the chemoattractant. This here is our blank side, where there's only very few ones present. They spend only very little time, and they spend a lot of time around our chemoattractant. And it's repeatable across multiple devices. We had we repeated this experiment multiple times. So we've got the beginning, 15 minutes, 30 minutes, 45 minutes when they're moving randomly allowed. And we can show on the heat graph that they like to sit close to our chemoattractants. We did the same thing with the planaria, but then we didn't use an amino acid solution as a chemoattractant, but a liver extract. You can see a little bit of gross stuff sitting in the reservoir here, and that's where it becomes clear why we like to have a membrane in there, because we didn't want to have bits of liver floating around during our assay, just in case our camera would be thinking they would be a planaria. Experiment otherwise is exactly the same. We lift up the little reservoir. They're just hiding in the side here, but the planaria are in here. Here you can see swimming around the food source. After the 60 minutes, we get our yummy stuff from the liver distributed all through our chamber, and we can see the planaria also being present in the bottom. Again, our heat map is probably even stronger than we saw with the zebrafish. They spent a lot of time close to the food stuff, because that liver stuff must be really, really, really nice, and I would want to be close to the food. And only very little time of the planaria was spent down in the bottom with our buffer solution as a control. So this shows that with integrating a little membrane, we can make a device custom and customize it and still have it small enough. So these in here are 5 millimeters, so this is a 4 centimeter scale. It is still small enough to observe what's going on with a zoom microscope. Because that's one of the problems that of studying these aquatic organisms. They're relatively small, so you need to have small tools in order to study them. And with the microfluidic devices, or we should probably call this fluidic, because it's not really microfluidic anymore, we can actually contain them in a small area, and we can see the little critters, the little planaria, yummy, swimming towards the food and not towards the buffer. Last part. So looked at print pause print method for chemotaxis. And our last thing was when we start playing with the printer settings and we applied a grayscale mask. So this is a couple of slides from Harry, and I couldn't remove the background. That's where why we have all the bubbles in the background. I'm not sure I reapplied the current slide format that I used 5 times, and I couldn't get rid of the bubbles. So bear with me. The rest of today's lecture's got bubbles. So what we've done with Harry's PhD, we started, and that's in 2013, people did post-print integration. So with Harry's, we started off with printing the porous materials post-print. Great post-print. So what we're now going to be looking at is in-print integration of fluidic, of different porous materials. So we don't have to touch the printer anymore while we're printing. The way that we do that, propose to do that, is by introducing a grayscale mask. And we're very pleased this work was just accepted in small structures, which got an impact factor 12. So we're very happy that that happened. So instead of applying homogeneous light through our DLP printer, we applied a grayscale mask. So pretty much what we did is we locally put on some sunglasses to reduce the light intensity, reduce the dose of light that our polymerization mixture got. If you've paid attention to any of the other optimization we've done, we've always played around with the polymerization kinetics to tune the porosity of these polymer monoliths. Here we're doing it not by changing the resin chemistry, but we do it by changing the light dose that individual parts of our print get by putting some artificial sunglasses on top. So we can see here, no sunglasses under the materials that under the conditions that we make. We get a dense material being formed. Subtle sunglasses, we get some level of porosity. The darker our sunglasses get, the more porous the material gets as well, because the light dose received is smaller. How do we do it? Grayscale mask is something that's a function in most of the 3D printers. So you just upload it when you've sliced your images for projection. You merge the grayscale mask at the locations you want to apply that in the G-code file that goes through the printer. So printers that allow you to apply a grayscale mask give you an option, do you want to insert a grayscale mask? And then you say yes or no, and we said yes. This is the way that a grayscale mask that we use for developing our conditions look like. And as you can see, they are just like sunglasses. We've got ones that are completely transparent, and they get increasingly dark gray up to the point they're completely black. If we look at the pic on the different structures that are being formed, we get nice polymer structures formed from GS0, so no grayscale applied, all the way to about GS75, after which our light dose is smaller than the critical dose. Think about our previous lecture. And we don't get any real structures being formed anymore. So this is our playground. Lots of animations in here. So increasing gray mask intensity, no sunglasses, dense, dark sunglasses, little dose, porous material being formed, but the dose is still larger than the critical dose, so we still get a structure formed. So we should be able to print a gradient in porosity when we have a gradient in our grayscale mask during our print. If we do some structural characterization here of analyzing the electron microscopy images, we can get a kind of an idea of the distribution and the average pore size. So that's what we've done here. If we've got a grayscale of 75, our apparent pore size is in the order of 50 nanometers. 65%, it's a little bit larger, and 55, 45, we get smaller, and we get at 45%, so not very intense pair of sunglasses, we get lots of energy, our dose is relatively high, and we get a narrow pore size distribution being formed. Same thing here. If we decrease our grayscale mask, which means we increase the intensity, we get increased geometrical density and a decreasing porosity in our material. Interestingly, if we compare that with a resin that didn't contain any porogen, our porosity is fairly similar, but we do have a difference in our density because our non-porogen containing polymerization mixture contains more monomer and therefore gets a denser polymer being formed than the one that has contained a volume of porogen in there. So what did we use this for? The first thing that we wanted to look at to see if the fluidic properties of the materials printed were any different. So we had a test device here with a couple of reservoirs to put the liquids in. We applied grayscale masks where we had no one where we printed with a 0% grayscale mask all the way up to 75. So we would expect a dense material to form here because it was fully blasted at a very high dose, and we'd expect increasing porosity going to the sides. Indeed, if we print these devices, you can see that with the dense material, the fluorescent solution doesn't wick up because it's dense, there's no space for the water to go. The more porous the material gets, the bigger the pores, the more transport of water. Yehey, could graph that. So you can see that initially, short distance traveled, we get fast transport of water. The deeper we go into the pores, the longer the length, the higher the flow resistance. So we get a little bit of tapering off. So this all works like we expect as a fluidic person. We then wanted to do some analytical chemistry because most of my life I've done analytical chemistry, so let's do it here as well. So we had our design device where we wanted to have a porous membrane in the middle separating two fluidic chambers. The way we planned to achieve that was by applying a grayscale mask in the middle. So 3D printing of our device and our grayscale mask. And this is the way the device ended up looking like, where most of the device, the bulk material, was printed with GS0, resulting in a dense material that shouldn't be wicking any liquid. Whereas the membrane here was printed with our maximum grayscale, with grayscale 75, for the creation of a porous material. So here you can see a photo, and you can see that there's a slight difference in the appearance between our highly cross-linked and our less cross-linked materials. And if we look at that a little bit more close, we can actually see a step, because with the high dense, we get a little bit of overpolymerization. So these are optical profiler images where we look at the scattering, the reflection of light, and we can see that there's a tiny step going down from our fully exposed resin down in a reservoir You can see it at the 200 micron scale. We can see particularly at the interface, we get a little bit of overpolarization. This one shows it the best. You can see that there's almost like a doorstep being formed here. That's because we don't get complete, or we don't get as complete polymerization happening where we apply a grayscale mask than is happening here. We have lots of radicals here because we apply a really high dosage of light, which means that the monomers that are just diffusing around here, that are not being consumed in the polymerization reaction with the high intensity grayscale mask, they are consumed here. So we just get in the top layer a bit more polymer being formed because it's available. However, we looked at the SEMs and we saw that in the transition zone, there is actually that thickening of the layer only happens in the very top one, because in all the other ones, the layer height restricts that overpolymerization, because you can't form a polymer when the previous layer is in the way. We can see at GS0, we can see the printing in a layer by layer manner. We can see that's there, but we also can see that we've got good bonding between the layers. Same thing from the GS75. We can distinguish the different layers, but we can also see that they're well bound together. And that doesn't change in our transition zone here, where we've got similar layer height, very similar structure. Looks like a nicely integrated device. So we applied that to an ion assay. So what we did is put a soil slurry from a mining site on one side of our device. We put an ion or a phenanthrene solution at the other side, and phenanthrene forms an orangey color whenever it's in contact with iron. Here we've got images of the membrane that's sitting in the middle, and that's where the ions from the soil slurry diffuse into the membrane, and the phenanthrene from the solution here diffuse also into the membrane, and they meet each other. If phenanthrene sees iron, it says, hey, I become pink. And we can see here the intensity of the pink band where the two different solutions meet and one another does increase with the concentration of iron in solution. Here we can see a nice calibration curve, and we were very pleased to learn that our quantification using our microfluidic device, using a digital camera for imaging, was within 2% from in ICP-MS analysis in our reference lab. So we got very good analytical data out, calibrating the color intensity of this colored line in our functionally 3D printed integrated device. So functional integration of porous structures by digital light projection printing. The handles that we have to manipulate are the resin chemistry, the monomer itself, which determines hydrophilicity, hydrophobicity, the porogen that you can add to add porous properties, the photo initiator and the photoabsorber that they can use to play with the polymerization kinetics, and in that term, play around, manipulate the pore size to tune them to your application. Similarly, the printing parameters can be used to tune the porosity to your application, and we've played around with the layer number and layer height to get big pores for the chemotexture studies, and we've used the exposure intensity using grayscale mask to get in-print porous structures. This is the group that did it. So Suleiman, a PhD student, was involved in developing the control system for the pressure system. Rich is our engineer that makes all our instruments work and keeps our vacuum pumps and everything up and does all our CNC milling. Here we've got Harry, that is the PhD student that has been the lead and the driver between all of the research that I've presented in the past hour. Sumin has done the PCR analysis, so she's doing the DNA extraction work that we may be seeing a little bit more of tomorrow. Dan worked as a postdoc. She's just left to go to another university, but she's been involved in supervision as well. And March is now left to go somewhere else, but she did some particle separations using the small 3D printed structures that I showed you. And that's me in the tree. So thank you very much for listening. And I hope you can see that what the mechanical engineer can achieve in optimizing resin chemistry. So whatever you're trained, you can always learn a new trick. Thank you very much. And any questions are always welcome. Any questions? Were our biotech people happy to see living things? We'll be getting there. We like to play around, but it takes a long time for zebrafish to actually get babies. We tried for 2 weeks, and then only then we had some embryos. So biological experiments are much more temperamentful than just getting chemicals off the rack. But they're good fun, and the planaria are just cute to look at. Thank you, and enjoy your evening. Bye. Oh, nee. I still have coffee now.